I'm going to take advantage of the opportunity to talk to you all and really give you a couple of pieces. I want to talk about my topic here, some things we're doing with mechanical imaging, with magnetic resonance elastography, with MRI measuring mechanical properties of the brain. And we'll tell you what hippocampus is, and maybe we'll even tell you why we call it Jellyroll here too. But I also want to give you an update on all the exciting things that are happening in MRI on campus too. So as being on campus, you should be aware of these things. And you know, I think it's a really exciting time, despite the fact that not a lot of other things can move forward. There's been a lot of activity in the MRI space. So I'm going to spend the first part of my talk telling you about those exciting things, and then we'll get into the science part later at the end. So it's a really exciting time to be doing MRI at Illinois. The Biomedical Imaging Center in Beckman Institute has two state-of-the-art 3T human Prisma MRI systems. One of them is multinuclear, so most of the time we're imaging protons with MRI, but one of our systems is multinuclear. Prisma is still the premier 3T system, and so we're glad we have two of those. And then we just got a new 9.4 Tesla preclinical animal system, which I'll talk a little bit more about too as we go through. That just came in June, and then we have a new 7 Tesla human system that just came to Carl in September. And so a lot of things are happening and a lot of construction projects going on. MRI is on the rise in town. Carl Hospital, over the pandemic, when radiology slowed down, they upgraded their 3T MRI scanner to a Prisma. So now they're actually really compatible with us. And so people like myself and Fan can go back and forth really easily now because the version of the software is even matched. And there are 3 additional scanners coming to Carl, which are going to open up a variety of other opportunities. In addition to the 7T, they're getting a couple of other systems that'll be compatible with what we can do here. VetMed has a 3T Skyra, and Fan Lam has been running things there too on that system. And then there's even a 3T Skyra at Christie Clinic out at the Fields, Carl at the Fields, which is exciting too. So lots of interesting pieces of equipment. When I got here, we had a head-only 3T MRI scanner, and that was the only 3T in town. And now we have lots of equipment. So, so Carl Hospital is also growing people and resources too. They're getting a new imaging center of excellence, and they just hired Bruce Damon from Vanderbilt. And so we're really excited to have him come and join us. And so, you know, not just the equipment, but bringing people, bringing collaboration, and really hoping to drive the future of clinical imaging at Highfield. This builds on our excellent history, and I want to point this out because exciting things happening soon along these lines too. So a lot of you know that Paul Lauterbur was here on campus, and he won the Nobel Prize in 2003 for the invention of MRI. And he was a professor here from 85 to 2007 when he passed. And we had a great Nobel Prize symposium and celebration in 2003 when he won. He shared the Nobel Prize with Peter Mansfield, who was a postdoc here in the 60s too. So almost everybody involved in the development, the commercialization at GE. So please, if you have questions about that, let me know. But so many of those people came from Illinois, passed through Illinois at various stages of their career. And so we really feel like, you know, there's something in the air here with the resonating protons. So it turns out that Paul invented MRI sitting at a burger joint, wrote it down September 2nd, 1971. And so 50 years from there will be September 2nd, 2021. And that's why I wanted to talk about this too, as a good opportunity to celebrate the 50-year anniversary of the invention of MRI. He wrote down, these are things that are available online. This is one of the pages from his books that he wrote down, his notebook, September 2nd, 1971. He had somebody notarize it the next day, and he explains how the imaging would work. And then he spent his time in the lab from September 2nd to October 10th when it actually worked. And we have found the data notebook from that. We just found it 2 years ago in the summer. Looking through a storage shed, I found the original data notebook from back at that time. And with the time frames of 2 and 3 a.m. on the pages of when he was doing the experiments. And so, so here is one of those data notebooks from October 10th, 1971 when it worked. It's very similar to the Nature paper that came out in 73, this data, that there's a couple of tubes, you measure at a couple of different angles, and you get a projection image. And so we have these actual notebook pages, and we have a new museum on display in Beckman Institute with the Nobel Prize and some of the pages of this book. So, so this is Paul sitting at one of his human magnets, probably about 76, 77 in the basement at Stony Brook. And we have found these magnets, and we found a lot of the peripheral equipment and we put them together in a display in Beckman Institute. So we have his spectrometer he's sitting at. We even have the RF amp that was sitting up here. We found that too. Power supply cabinets. And then we have the world's first human MRI scanner from 1975. And that one was a mistake. And we have the one he got in 76 and January 77 delivered here too. So we have the world's first human MRI systems. And so we've been working to try to celebrate this anniversary of the invention of MRI. And so given the pandemic, haven't quite closed the deal on making sure that we're going to have a big party yet. So, but hopefully we can figure that out, and hopefully things can turn around by the end of next summer. I wanted to show you some of the new pieces of equipment here. This is our 9.4T Bruker system, a preclinical animal scanner, 30-centimeter bore, good for rats and mice, and even bigger than that. We had it delivered in June. It took some doing. We had lots of things go wrong. I'd be glad to talk about those to people some other time, but August 26th, we got it up to field, and it's been working great ever since. We have some training going on. Our first scans were on a tomato. I have pictures of that here. This is Mr. Tomato that got scanned. These are really great images then you can see. And then just recently we had Shreya Majumdar, who's our scientist at BIC, and this is his net ID if you want to email him. He is helping people get ready and get up and running on the machine. And we got some really great images of a rat working with the Gillette lab. So it was a deceased rat at this point. We don't have all the other animal components hooked up yet, but we're getting close. So, but great images, something like 150 microns and plenty of SNR to work with. So we're really excited about that. 7T human MRI system got delivered September 10th to Carl, and is being currently assembled. We get to start training at the end of November, and we get to take over in December for handoff. And we hope to start running subjects in January. So if you're interested in this, or if you're a student and want to get scanned at 7T, how many people have been ever even scanned at 7T? Not too many. We're going to be running a really big study starting in January, where we hope to scan about 200 people twice, 1 year apart, and look at how much more information you can get out of 7T than 3T. And we're going to have a really great facility. It's under the Heart Vascular Institute at Carle Hospital is where this is being put in. There's nothing down there right now. It was just a whole bunch of rocks in the basement and an unfinished basement, but we're going to have a really nice suite with a great control room, huge magnet. This thing is a beast, but really nice space and subject prep areas for clinical or research. So really excited about this. We were going to have a door that actually was transparent and then became opaque, but we're going to go for really good shielding instead. So we're not going to have too many bells and whistles. So that is an overview of why, I'm really excited, and also why it's a really busy time for me. We have a lot of stuff going on, but really exciting opportunities as we start to build things. I want to tell you a little bit about what my group does, and then we'll get into the MRE components. And so what we do is we design MRI pulse sequences and image reconstruction approaches to try to measure certain aspects of physiology that may not be possible from the current technology. And so we try to develop new technology to really push the boundaries and be able to do things that wouldn't otherwise be possible. And so I have two good examples of this here from two of my bioengineering graduate students that are doing a fantastic job. And so we have some great results to show. So I just want to show really quickly, one of those is fast imaging. So we work with a lot of groups that do speech imaging, looking at cleft palate birth defects, and looking to see how the muscles develop in different populations. And MRI is very slow. It takes seconds to get an image, or if you push things, you can get several images per second, but usually very limited coverage. But working with Fan and with Jipei Liang and others, we've managed to develop technologies that allow us to do full 3D imaging in real time at hundreds of frames per second. And so, and we get really good quality images. So I'm just going to show an example here. I will warn you, I've shared the audio, and there is audio on this. And if you recognize the voice, this is one of our graduate students here. So.
Bye, baby, a bit. Bye, baby, a bit. Bye, baby, a bit.
And so we're usually interested in looking at the velum. You can see it kicks back to shut off the nasal cavity from the oral cavity.
Bye, baby, a bit.
So, being able to get that high of a frame rate to be able to see these muscles contract and where they move around and be able to model the vocal tract functions. And then we're going to be looking at kids, cleft palate. looking to see how we can distinguish the dynamic aspects of that. Right now, if you were to get an image related to those, it would just be a structural image, and you have to stay very still. Another project we're working on is really trying to measure very slow flow. So this is Riwei Jin, and this is Mingjiao Zhang. We're trying to measure very slow flow. So in this case, we would have flow. So we're looking at tubes that get embedded in usually pediatric populations because their CSF, their cerebrospinal fluid, doesn't drain properly. And so a shunt is put in to provide a pathway to drain that out. But 50% of those fail within 2 years. And the way you find out is people come in with neurological symptoms and their ventricles are big because they've built up pressure. So we're really hoping to be able to measure the flow and be able to see. Somebody could come in for an annual checkup and they could determine that their flow was starting to become impaired before they actually had problems. And there's no other way to do this. It's really hard to measure this flow because in 5 minutes we get 1 drop. So we're trying to measure flow with MR at that flow rate, about 0.2 milliliters per minute or less. And so we get 1 drop in about 5 minutes. And so what we've been doing so far is working with Carl. Whoops, sorry about that. We've been working with Carl on extraventricular drain patients where they have tubes embedded in them due to trauma. They're in the hospital, they're usually unconscious, and they have tubes embedded in them, and they have a tube coming out the top of their head, which drains into a bag. And we're able to then measure this very slow flow rate, but then also confirm what's coming out in a bag. And so this is showing you an example of one of those tubes coming out here that's embedded in the cerebrospinal fluid. So, trying to do things that are, beyond what was possible before. So what I'm going to tell you about in this talk is something that we started doing quite a few years back, but have really continued to develop the technology on, and that is trying to measure the mechanical properties of the brain and trying to understand how the stiffness of the brain affects its operation and how important it is for how the brain is put together and the structure of the system. And so we're going to tell you how we make those measurements, and then we're going to show you some examples of the type of information you can get. We're going to show you that we found that even in healthy young adults, this technique is giving us information that we can't get in any other way. It's actually predicting brain performance on particular tasks in a very sensitive manner. And so hopefully we'll convince you of that at the end of this. So I like to start by talking about this is such a big project. In fact, I need to add more names to this now. In the last couple of weeks, we started some more collaborations, and in particular with the Anastasio group to try to automate some things, which I'll talk about at the end. But it's a big project. We have a lot of help. We have people who are specialists in memory performance and those very sensitive memory tasks. And we have people who are working on epilepsy and trying to give us nice behavior where there is some expected changes in stiffness. And then we have people working on sequences and inversions and mechanical inversions and all sorts of parts of this process. And so it's been a very big collaboration and very exciting and moving quickly. So why measure mechanical properties in the brain? What would lead us to this? There are some good reasons to expect that mechanical properties of the brain would be very important, and especially on this campus. We have people like Taher Saif who are looking at the mechanical properties of neurons and how neurons under tension have different behavior than neurons that aren't under tension. So there's a good reason to think that the mechanical structure of the tissue does have a dependency on how well the brain functions. And then also cortical folding work and a lot of things over the years have really pointed to the impact that mechanics have early on in brain development and organization. So we started looking at this, but then another thing became obvious too. If you start looking at the orders of magnitude of contrast that are available in an image, mechanical properties vary over several orders of magnitude in the body too. And so that gives us something that potentially has a wide range that can give us a great deal of sensitivity to microstructural organization in the brain. And so we're really interested. We think that mechanical properties will tell us things about myelination of axons and axonal integrity, how those axons are put together, and whether whether that myelin layer is fairly continuous throughout the organization. Axonal packing, how densely do the axons get packed? Do axons running in different directions packed together? And then also something that we've been trying to get information about too is inflammation. And so as different parts of the system have inflammation, does that should change the mechanical properties of the system? And so we're hoping that become a sensitive measure of inflammation. So we started thinking about how to do this. And our initial ideas were quite crude. I'm going to tell you, if you wanted to measure, brain deformations in the scanner to try to see how the mechanical properties are, you might think about it this way. You might think, I want to cause an impact to the brain and then measure how much it moves. And so we actually have a senior design project with mechanical engineering students that we did that did something not quite this bad. What we did is we dropped people's heads a small amount and had them hit the scanner bed, and it was supposed to be random and you can't predict it. Otherwise you tense your neck muscles and not let your head fall. And so we did these things for a while, and here are some images of that. And so of course this is probably me in the scanner, but we do something called tagging, grid tagging, spam. So grid tagging, and you can apply grids at a particular point in the image. I have no idea why my video did that, but it looks like we're not going to get to see it again. Oh, there we go. And if you actually look, these are the ventricles, which are filled with water. And so I'll play it one more time, and you'll actually see that the top of the brain moves one way and the bottom of the brain actually moves the other at a particular point in time there. So you're able to actually measure displacements and differential displacements here. You can estimate those. You can measure them in different planes, and you can get quantitative measures of velocity through this technique as you do that. So you can see it bounce and oscillate around here. We didn't exactly hit straight on, so we had some rotation there too. So we were doing this in order to get a full experiment done. I think we had to do about 60 drops per person, but I assure you we did all the safety components with IRB to make sure that we were not causing much of an impact. So these are definitely exaggerated arrows here showing velocities. It turns out that in the late 90s, some other technology was developed at Mayo Clinic that starts to make this process easier. And it is the development of a resounded device, which then generates shear waves to propagate through tissue. And it's been used a lot in the liver. And so instead of having to impact people, punch them in the, in the rib cage over and over again, this device can vibrate at a particular frequency. And you can image at steady state then. And you can look to see how these waves propagate through the tissue by changing your offset and sampling across that steady state oscillation. You can sample at different time points. And so this allows you to really investigate tissue in a very reproducible manner and a very comfortable manner. So this thing vibrates very little bit. And for the liver, you put it on the rib cage and you would be able to image then that vibration in a very repeatable manner over and over again. So this allows quantitative palpation. And that's what magnetic resonance elastography is. So elastography, we're going to get quantitative maps of the stiffness. And so if you think about it, if you went in and you were complaining of pain, one of the first things a doctor might do is feel for stiff inclusions, and that would give them an evidence of whether there was pathology in that region. And so what we're doing here is we're using imaging then to measure displacements that we're applying from these vibrations and to be able to feel, to be able to image and see if there are stiff inclusions. So MRE is quantitative, specific, localized, repeatable, non-invasive. It's pretty comfortable. And it's been used a lot in liver disease. And so this is showing a couple of different levels of fibrosis of the liver. And these shear waves, the wavelengths get longer as things get stiffer. And so you can see then very clearly how things are progressing here across a set of patients. And then we can actually do some inversions in order to be able to turn this into stiffness maps. And you can see the stiffness, and we can measure that in a quantitative unit like kilopascals. So at Mayo Clinic, they've been really pushing liver MRE and its reproducibility, and they've actually gotten to the point where they can replace invasive biopsies. So if you were to do a biopsy early on and you were looking for early stiffness, you would have a sampling problem. You can only take a few samples. If you didn't sample some of the regions that were stiffer, you might get an incorrect picture as to what's going on in this liver. Okay, and so with imaging, you actually can sample the whole thing. You can see it all. So I'll tell you a little bit about how it works. And this is just looking at those shear waves propagate through an agar gel phantom. And we have a stiffer component of the gel in the middle, and you can see that the wavelength gets longer, the propagation speed gets quicker through that tissue or through that gel as opposed to the stuff on the outside. And then we can put it through a inversion algorithm in order to get an estimate of the stiffness then based on looking at how these things propagate through. So a typical MRI experiment, we need an external actuator. So we would have that vibrating pad placed up here. We do phase contrast imaging. So we get a phase image related to displacement. Very sensitive displacement though, because the brain is only going to move on the order of plus or minus 5 microns. So we need something that can be very, very sensitive to that displacement level. And then we need an inversion algorithm for property estimation. And this is what we've worked with Aaron Anderson here locally, who came from TAM and mechanical engineering. And we also work with Dartmouth on some finite element based mechanical property inversion procedures. So, and those are actually computationally quite intense. And so we use a lot of high performance computing in order to get that. So we have a resounded system, and we have, instead of putting a big old drum on the side of the head to cause vibrations, we have a nice little pillow that goes behind the head, and it inflates slightly. And then this is equivalent to a subwoofer, a very, very expensive subwoofer. Anything with MRI is very expensive. So this is anywhere from, say, 50, 60, 80,000 up to 120,000 if you want the software too. And then this sends these sound waves through. And they vibrate this pillow, and it's very, very reproducible in terms of its oscillations. And we then run an MRI sequence where we line up with these oscillations. There are trigger pulses going back and forth between the MRI scanner and this, and we can line those up, and we can encode very small displacements. We try to do 3D displacement. So you can, I'll show on the next page, you have to encode all displacements in all 3 directions. And we try to push the spatial resolution and get full brain coverage to help us with our inversions. And then we can get a nice data set in about 6 minutes. So it's very short. Okay, so with the pillow behind the head, these are the types of displacement maps you would get for the brain. And the scale here is plus or minus 5 microns. And so we get a lot of displacement in the head foot direction. If you have a pad behind your head, it's causing you to nod your head, you know, and so we get a lot of head foot movement. We got a lot of anterior, posterior, front back movement associated with that, but we don't have a whole lot of left right movement there. Okay, and so because there's not a whole lot of shaking in that direction. But there are some new things that, you know, Aaron Anderson is doing where we can shake in different directions and we can shake at different frequencies and things like that in order to put these pieces of information together. I want to explain what you get out of this and kind of how to think about these elastograms and what we have learned about them, and then I'll give you a couple of applications associated with this. So this is just an axial slice through the brain. So just cut your, you know, through your brain this way, you know, left and right brain, and we have the corpus callosum outlined in red here. These are the main white matter fibers that are connecting both sides of your head together. And then we have these blue fibers, which are chronoradiata, which are fibers that run up and down your head. So they're going to be connecting the top parts of your brain to your muscle network below for controlling muscles. So these are really large fibers too. So 2 really large fiber bundles, but they're going to have some very different properties. And this is a stiffness map associated with them. This is an older map, but you get a higher stiffness inside these white matter structures than you do elsewhere. And then this is a different piece. So when we do our mechanical property estimations, we get 2 things. We get a stiffness and we get a loss. So we get a storage modulus and a loss modulus. Storage modulus is really related to stiffness, and when you deform something, how much that energy is going into the deformation and coming back. And then a loss modulus is how that energy is dissipated as those waves propagate through the tissue. And so I'll explain why these are different. They're actually quite different, and that tells us a lot about the structure of the tissue. And then I'm going to show you a diffusion image here too. Diffusion is a common MRI technique where we can encode the displacement of water in different tissues, and we can look and see which way diffusion is restricted, okay? And so if I'm inside an axon, I can't really diffuse outside that axon. My water diffusion will primarily be along the direction of that axon. And so red here is right, left, blue is up, down. And so these are showing us that the water in the corpus callosum here is primarily restricted to diffuse in this direction. And these are primarily restricted to diffuse in the head foot direction, going, you know, up, down. And so that allows us to see where these large fiber bundles are. You can't actually see the individual fiber bundles in a normal MRI image. You need to do something else. So, okay. So that hopefully gives an overview of what we're going to be looking at. So we want to go through and look at these 2 things, and then we're going to look at gray matter, stiffness, you know, or damping ratio. We're going to look at in the next slide, gray matter, white matter, corpus callosum, and chronoradiata. So white matter, but then very specific large white matter fiber tract and a very specific large white matter fiber tract here. So, so we get stiffness out of these estimates, and this stiffness is really going to reflect axon properties and organization of the system. And high alignment of axon leads to higher stiffness, and larger axons lead to higher stiffness. And so this is from the diffusion map. So looking at tracking here, and the corpus callosum and the corona radiata. Whoops. So if we look at the stiffness, okay, of these things, we can see gray matter is very different than the three white matter components. Can you see my cursor, just so I know if I'm waving at something and you can't see my cursor?
Yes, I can see that.
Okay, great. So gray matter is less stiff than white matter. And then even among these white matter then too, corpus callosum are these really large fiber bundles connecting side to side. They're very tightly packed, and because they're all running in the same direction through this, and they're higher stiffness than regular white matter and then corona radiata. So we get some information about, you know, the axons and the organization of the tissue just by looking at stiffness. And then it turns out we get out a lot more information by looking at this loss modulus. So the dissipation of that energy as things propagate through the tissues gives us a lot of information too. And so you can see that here. It turns out that in the corpus callosum, we have a whole bunch of fibers that are oriented perpendicular. They're oriented in the same direction. They go across in between the two hemispheres. And when we're putting energy in, they're kind of like rebar, and they just transmit that energy pretty efficiently. And so the loss modulus is actually pretty low associated with the corpus callosum. But the corona radiata are fiber tracts that sit more on the outside of the brain, and they're intersecting with other fiber tracts that are running in other directions. So if you can imagine, we have fiber tracts running in one direction, we have other fiber tracts that are intersecting those perpendicularly. And as we put energy in to vibrate that system, there's a great deal of degrees of freedom there, an opportunity for dissipation of that mechanical energy in those interactions between the fibers. And so we can see here, corpus callosum has a really low loss modulus, and corona radiata has really high loss modulus. And so we think this is a really important part of the information we're getting out of these mechanical properties. And we actually turn it into damping ratio, which is something if you're doing normal dynamic systems, that's something that might make more sense to you. So we do damping ratio instead, and you can see here corpus callosum really low and corona radiata very high. And damping ratio will become important as we look at this later. We've looked at repeatability to make sure that the measures are repeatable. And so we measure people over and over again. We're currently doing a repeatability study over a longer term time and looking to see how things repeat. We have a couple of experiments that we're working on where we scan people multiple times over the course of some physiological event. And we really need to understand what the reproducibility of the measure is across those. And we're finding really high reproducibility. So this is older methods, and we still had coefficients of variation on the order of 5 to 8%. But I think our latest numbers are 5% or less. So, all right, I'm going to give you, so hopefully you kind of understand what we're looking at. I'm going to give you an overview of some of the application areas that we've looked at and some of the interesting things that we're finding. And then I want to talk a little bit about the technology required to get there and where we're headed as we go through this. So when we came into this field of MRE, the techniques were not very localized. And so this is an example of some work that was published a while back, using MRE to look at brain properties. And what they were doing is splitting the brain up into inner, outer, frontal, dorsal. And so big regions where they could try to take averaging and try to get mechanical properties. With our techniques, we've been able to increase the spatial resolution and localization of these, so we can actually get really high quality images associated with mechanical properties. And we can split up into specific regions of the brain. So this would be looking at the subcortical volumes. And so amygdala, hippocampus, caudate, putamen. And we scanned 20 college-age kids, and we were able to see that there are differences in the damping ratio between different tissues based on their structure, and that they were actually highly reproducible across a set of healthy adults. There are age-related changes. We've been doing a couple studies to look at this. We recently got an R03 funded to look at age-related changes here. And so as you get older, your stiffness is going to go down. And so, and it goes down at a measurable rate. And we're going to look and see if that becomes a more sensitive measure than other things. As you get older, your brain also shrinks. Your cerebrospinal fluid spaces also get bigger. And so we're going to start to look at the differential sensitivity across age associated with MRE. So one of the exciting results we had very early on that really helped establish this as a sensitive method was a collaboration with Hilary Schwarb and Neil Cohen. And looking at, they have a very sensitive measure of memory and memory performance, especially relational memory performance, which is where your brain puts two components of information together and links that information together, okay? And that's really important memory and used quite often. It primarily runs through the hippocampus as controlling that. And so they have this great, very sensitive measure that they can even see variation in healthy young adults. They can see mild cognitive impairment. They can even apply it in Alzheimer's. So it's got a wide dynamic range of sensitivity, and we were happy to pair up with them. And so we ran a study in healthy young adults, and we looked at memory performance related to MRE. And one of the things you might do if you were just running this study and you didn't have MRE available, people look at volume, the volume of the hippocampus and how it relates to your memory performance. And it turns out in older adults, as you get older or looking at old versus young, volume of the hippocampus becomes a big determining factor of memory performance and predicts a lot of the performance of an individual. But in healthy young college-age kids, it's not a very good predictor of memory performance, okay? So just looking at volume in healthy young adults, it doesn't predict. But we looked at damping ratio in the MRE measure, and we saw a really huge predictive value associated with MRE. So if you look at this, an R of 0.7 means the R squared, the amount of variance we're explaining, is about 50% of the variance in the data is explained by the MRE measure. So by measuring the damping ratio of your hippocampus in healthy college-age kids, we can predict your performance on a hippocampal dependent memory task. We can predict 50% of the variance of that across the population. So it provides a very sensitive measure. This was a really exciting result. We've reproduced this result in a couple of different populations now. And this has been our go-to task. And it's interesting because it also was our first task. So I think there's a lot of stuff out there that we haven't done yet that will also show some really interesting relationships between the mechanical properties of the brain and how the brain works. We might wonder from that data set, in fact we did, as to whether those kids that performed worse on the memory task and had a different damping ratio, whether they actually just have a different damping ratio of their whole brain, or whether it's something very specifically focused on that task. And so we ran another study right away, looking at two different cognitive tasks and looking at two different regions of the brain where they are. So we looked at fluid intelligence, so more of a generalized intelligence measure, and we looked at orbitofrontal cortex, where that is primarily located, and then we looked at hippocampus and relational memory again, okay? So, which is what we did on that last one. And then we looked at the damping ratio of those regions. So we matched both of them. So you can look at the damping ratio of the hippocampus related to fluid intelligence. You can look at the damping ratio of the hippocampus related to relational memory. And so that's what I show over here. So you have hippocampus damping ratio on this axis. And if you're paying really close attention, first of all, I'm impressed, but also I want to explain. This says damping ratio prime. Our cognitive neuroscience collaborators really want to see positive correlations. And so this is actually 1 minus damping ratio. It's inverted in sign so that higher is better. That was determined that would help us out. People would understand it better. So higher is better. And then this is the performance on the memory task, and higher is better. This is a performance on an executive function task here, okay, that really stresses that fluid intelligence system, and higher is better. And as you can see, hippocampus damping ratio is related to memory performance, okay, and very significantly so. And hippocampus damping ratio is not related to executive function, okay? And likewise, orbitofrontal damping ratio is not related to memory performance, but it is highly related to executive function. And it's really interesting. These are very similar relationships between these two sets of data, different parts of the brain and different performance between the two groups. So it's not just a global effect, it's a very localized effect. It's actually functionally specific and very exciting. So we've looked at a couple other things. I think I will skip this one, just trying to understand. We do a lot of aerobic fitness and memory performance and cognitive performance tasks here. And we're looking to see whether some of the things that we think that happen with aerobic exercise affect hippocampal stiffness and that is an indicator of the overall performance. And so does hippocampal stiffness mediate those impacts? And so still some work going on there. We've improved the spatial resolution of our technique to try to look at what part of the hippocampus is affected, is primarily related to the memory performance. So the damping ratio of what specific part of the hippocampus is related to memory performance. And so this is where the Jellyroll comes in. If you haven't seen a hippocampus, this is what the hippocampus looks like in cross section. You can see it here too in the MR image. These are separated up into different pieces. And people have been finding in animal models and the like that dentate gyrus, CA3, is where some cerebrovascular components come into play that predict memory performance in the long term too. And so we were really excited when we broke this up and we started looking at what is predicting. We found that the CA3 dentate gyrus actually was the best predictor of our memory performance. So here I have memory performance on the y-axis and damping ratio on this adjusted damping ratio on the x-axis. Okay, so we're really excited. This kind of leads back to some of the mouse model literature as to what might be happening here, and it really pushes us to try to get even higher spatial resolution. So we have something that's very it's a localized measure. It's very specific to the functional task, and it seems to be very sensitive. We've also been working with people at Carl Hospital, and in particular, Graham Huseman, who runs an epilepsy clinic. And in epilepsy patients, this happened early on, and we thought this would be a great test bed for applying our MRE techniques in epilepsy. One side of the hippocampus, one hippocampus will get some activity due to the seizure, and that can induce scarring in that tissue. And so that scarring or fibrosis can change the stiffness. And so we were really excited to try to look at this population and see whether MRE would provide a good measure of disease progression. So this is an example flare image in an advanced case of mesial temporal lobe epilepsy, which is just meaning it's primarily affecting the hippocampus here. And you can see in this flare image, you can see that this is brighter than this side over here. But it is a subtle, when you start looking at a lot of these, it starts to pop to you and you can see it right away, but it is a subtle effect. And so this is in the same patient, this is our MRE image, looking at the stiffness of the affected hippocampus versus the other hippocampus, the non-affected. And you can see huge difference in stiffness here between these two. And so we found that the difference in stiffness is quite large and very predictive. And so we kind of show that here. What might you do? You might look at the volume of the hippocampus. The hippocampus enhances and gets smaller. And so we can look in our control subjects, and this is ipsi to contra. Okay, so this is the affected one over the non-affected one, looking at the volume ratio. And you can see the volume is smaller in the one that's affected. This is the MTLE subjects, the epilepsy subjects, and these are the controls. And there is some difference there in the volume ratios. But this is the stiffness ratio, so taking the stiffness of this side divided by this side. And we can see that there's really good separation between the epilepsy and the controls here. And so this has us still very optimistic about what we can see. In the process of making these, we started looking, and we actually saw something interesting. We saw that as we expected, the epileptic side, as people go from control to moderate disease to severe disease, the epileptic side actually gets stiffer as we progress. But then something we were not expecting at all is it turns out the non-epileptic side is actually getting less stiff, and that this effect is even bigger. So this side is actually less stiff than a control, and that effect is even bigger than how much stiffer this side has gotten. And we see that across all of our patients. And so it turns out we think the body is reacting to what's going on, and it's actually affecting this other side. And it turns out as this disease progresses, what will happen is if it's not addressed, which may be hippocampal resection, if it's not addressed, it will progress to the other side. So maybe this is a good marker of how that progression may occur. So just wanted to give a quick nod to some of the technology that enabled this and where we're headed to as we look at having a 7T scanner come on board. I wanted to sort of, tip what we're going to do along those lines. We started this MRE technology because we were really working hard on making high resolution diffusion tensor images to really look at these white matter fiber structures. So this is what would be considered high resolution in the normal part of imaging and human connectome project like protocols. So for a 3T clinical scanner, this would be considered high resolution. You can see the corpus callosum fibers running across, corona radiata, and then a fiber that runs front and back here. And then I'm showing an axial view and I'm showing a coronal view too. So you can see the corpus callosum, which runs across. This is the two sides of the brain on either side of this image. And you can see those fibers, and you get up here where all these fibers come together, and you get what you would get in kindergarten if you mix too many colors together. You just get a brown mess, okay? But we've been really improving the spatial resolution and being able to see even up to these interfaces. And you can see these these corpus callosum fibers even just tucking up underneath these other fiber bundles. And this took a lot of work to get the imaging and the reconstruction right and get the signal to noise ratio up high enough. Turns out even here, I'll just point, we actually have really interesting signals in the cortex too. You can actually see cortical layers going through here too. So really excited about this. And this is going 2 millimeter isotropic, which 2 by 2 by 2 is 8 to 0.8 millimeter isotropic, okay? And so it's a huge reduction in voxel size as you go through. There are challenges associated with this. We scan and we get a huge amount of data, and it takes forever to reconstruct the images, like months, and then you forgot what you did and what was different about that experiment versus the other one, and then you have to go do it all over again. I won't say how many times that happened. So, but we've been leveraging graphics processing units. We've been working with great people like Fan Lam and really developing interesting code to be able to push things through quickly. This is one of them that we came up with Fan when he was a graduate student too. And so this is Impatient MRI, Illinois Massively Parallel Acceleration Toolkit for Image Reconstruction with Enhanced Throughput in MRI. You have to have a good acronym. We had a follow-up paper called More Impatient, which was great. And then we've been working on other code. This code is really fast, but requires a very focused person to be able to edit the code. Let's say you want to incorporate some new physics. It's almost impossible unless you have a PhD in computer science. So we decided to write, this power grid, which is more parallel computing for the rest of us. And so we've been doing that and making these things available and having people use them. And we've been using a lot of Blue Waters time in order to get this. So 4 million hours. The time for one data set for MRE to invert it with the finite element based method for even a moderate spatial resolution, 1.6 millimeter, takes about 1,000 core hours for one brain to get through there. And so we've been working on ways to speed this up. And how do you translate this? How do you deliver it to the clinic if this is the type of computation required? So I think there's some interesting challenges to solve still. We've been pushing the technology further. A lot of what I showed you was 2 millimeter spatial resolution, kind of like this, but we've been pushing the spatial resolution down to 1.25. So by 2 by 2 is 8. 1.6 by 1.6 by 1.6 is 4. 1.25 times 1.25 times 1.25 is 2. Right, 4 and 2, I'm pretty sure. This is, yeah, so this is 2. And so, so this is halving the voxel volume each time. And yet we're still getting plenty of SNR, and we're getting even higher resolution images. Aaron Anderson is working on something where we now stimulate at multiple frequencies, and we can jointly solve models at different frequencies, and we can get even higher spatial resolution associated with the measures. And then we started working recently with the Anastasio lab and Frank Brooks, and very excited by this too. There's one step in the process that's manual. I want to say it's undergraduate labor driven of masking brains, because the brains are phase images, and they have some very interesting artifacts at the base of the brain that are really hard to determine what's brain and what's not. And we can tell people and we can show them, but it's been really hard to write algorithms to automatically do that. And we just got results out in the last week or so from that. And we're really excited that this is our manual brain segmentation after we run the inversions. And this is our updated mode. And this is how the two masks agree, and they agree very well. And this is running them through and just looking at a quality control metric at how they run. So manual in blue and deep learning in orange here, and that they actually line up everywhere. And this blue line is our critical factor. If you drop below this blue line, the data is not useful to us. So we're really excited by this, being able to remove any manual step, being able to just pipeline it through some high performance computing and hopefully get results out the other side quickly. I wanted to say briefly why we go to 7T. So I think I indicated that a minute ago. 1.25 by 1.25 by 1.25. We got decent images at 3T. But it turns out the cortex is somewhere between 2 to 4 millimeters thick, and there's a lot of interesting things happening in the cortex. We need to be able to get 3 or 4 voxels across the cortex in order to be able to measure the mechanical properties and those shear wave propagations across. So 1.25 cubed is 2. We can actually get a factor of 2 in SNR from 7T to 3, from 3T to 7T. And so we're hoping that actually without much, even without any new tricks or magic, we actually can get the SNR we need at 7T to go to 1 millimeter ISO. And then we can start to push below that and really start to get 3 or 4 voxels across there, and look at things like cortical dysplasia or local stiffening of the cortex, which is actually really hard to see with any other imaging techniques. All right, so I'm going to conclude. MRE hopefully convinced you. It's a quantitative measure of tissue status. It gives us really rich information about how the tissue is constructed, how these axons, the integrity of the individual axons, their organization arrangement, and how they're interacting with other axons. And then even more important, it's actually sensitive to even healthy performance. So there's nothing else I can do with a healthy college-age student, put them in an MRI scanner, and have much of any predictive value of how they would do on a memory test. And yet with MRE, we're actually predicting 50% of the variance across that, which is amazing. So, so we're really excited by this technique. It has very high sensitivity. We're finding really significant relationships. I showed you all the functional tasks we've done. Everything has turned out to be very significant. We just need to do more. So, and MRE is a great example of how engineering neuroscience collaborations can quickly open new avenues of research. If we were just developing that technique on our own, who knows? But the fact that we show we can predict memory performance and that it's a very sensitive technique and we can start to look at populations where that makes sense, that's really what's made this technique work and what has given us a great deal of exposure for doing this work. So with that, I want to say thanks. All the people in the lab have contributed to this talk, and then I wanted to actually give you a picture of our 7T sliding into place. It's such a beast. They have to cut 17 inches out to drop it down below the floor even to fit it in a room. But hopefully then with this system, we're going to actually be able to unlock new things. And with that, I will end and take any questions.
Maybe I can start with a couple quick questions, Brad. Oh, first of all, thanks for the amazing talk. So a couple questions regarding the mechanical properties. So do you think what do you think the anisotropy of the mechanical properties could play in the role? And do you think it would be worthwhile to look into that just when people move from ADC to diffusion tensors? Do you think that's a promising direction?
Yeah, I think, this is one of the things we've been hoping to look at as we go forward. There is probably a great deal of anisotropy in these measures. Thinking about the rebar, even looking at the corpus callosum, and seeing how, when you have these things organized in a very particular structure, they have very particular properties. And so I think the orientation of that will be important. We're still trying to get through sort of getting the average properties and how things go. I think we have to get very sensitive measures to be able to distinguish and go into this in order to be able to answer these questions well. And how do you incorporate these things? We need new algorithms to run the finite element that can take into account the anisotropy. Another side of that too, though, it's not completely obvious how important it will be because the tissue has lots of points of mechanical interaction and lots of connections, and the glial cells are reaching out everywhere. And so it could be that some of the directionality is dampened as we go through and we start to look at this macro scale of displacement of shear waves. But I think it'll be a really interesting thing to look at. Yeah, so hoping to go to that point. And certainly if there are people with anisotropic mechanical inversion models, then definitely be great to make a connection and start talking about that.
So another question is regarding the translation to 7T. So from my experience is that a lot of things that you can ignore on 3T, you have to consider at a 7T, right? So what do you think are the major engineering challenges to move this to 7T?
Yeah, so because of the way we're imaging, some of the things that people ignore at 3T, we're actually addressing already. And I think that's why we're getting better images. When you do phase imaging, there's a lot of things that can cause phase. And so like if you wiggle your fingers in a 7T scanner, you're going to change the phase in your MRI scan, and your breathing changes the phase and all these things. But we're already measuring Navigator with phase in our data to correct it because we're doing multi-shot imaging. Most people are trying to get by with a single shot so they can just kind of throw that away. But we're actually measuring and correcting it already. So I'm actually hopeful that the small change from 1.25 to 1 is actually not going to be extremely difficult. I think pushing past that, we have trade-offs of time. The SAR is another thing we're going to have to worry about is we have spin echo sequences here and we're trying to go fast. So the heating of the patient we have to worry about too and things like that. So, but yeah, I'm actually hopeful that we're already measuring some of the phase distributions and correcting it. And we're doing that because in diffusion, it's really, really important. In MRE, it hasn't been as important, but at 7T, it will be.
Oh, someone sent to me privately, So thank you, Professor Sutton. Very impressive talk. I have a question about what is the relative ratio between the brain movement induced by shear wave compared to the brain movement induced by cardiac pulsation.
Yeah. Yeah, I think that's a great question. I think overall, brain movement due to cardiac pulsation has been something that came up again recently too. And the order of magnitude is probably a little bit bigger with the cardiac induced brain deformations than what we're applying with our device. There are some groups who are doing MRE based on the natural impact of the brain stimulation. You can actually try to encode when the brain is moving. People have done humming too and things like that, different frequencies to be able to get the oscillations, but there's this intrinsic mechanical stimulation from the heart on the brain, and people have been using that for MRE. We're running at a very quick speed. That's a very low speed. So I think in terms of the frequencies that we're sensitive to, it's actually not going to be picked up much by what we're doing because it's pretty consistent. And we do something called flow compensated encoding, which not a lot of other people do, but that flow compensated encoding will get rid of things that seem like steady movement during a scan. So, but yeah, I think it can be used to your advantage, and we're actually considering ways to use it. We have other problems where the low frequency is probably going to be really important, and so we're actually looking at doing that again. But yeah, I think the order of magnitude is probably bigger in terms of the actual displacements that are experienced from the heartbeat than it is from what we're applying.
Okay,
All right.
cool. Thanks. All right.
Yep.
Oh, sorry. Another quick question.
One quick question. Have you compared the mechanical property of hippocampus when patients are relaxed and using their memory? Yeah. So there is something called magnetic resonance elastography, functional magnetic resonance elastography that people are doing, where they're actually looking to see mechanical property changes when a part of the brain is active. We've done a little bit of that over the time too, and so you can imagine, the extra blood that flows into tissue might change mechanical properties. So there's reason to think it would be. It's a small effect. There's some groups doing it now. They have to be very specific when they encode it in order to be sensitive to such a small change. But I think there is something there. We've done some of the experiments ourselves with visual tasks, and the signal is so small and variable. We really didn't end up publishing any of that. We didn't feel very comfortable with it. So I think there is something there. It's just a really small signal. Maybe at 7T, that will pop and we'll be able to see it. And I think that'll be good.